Welcome to the PharmaSource podcast.
Today we're joined by Campbell Bunce, Chief Scientific Officer at Abzena. Campbell leads a global team of scientists spanning discovery, development and GMP manufacturing, and is responsible for ensuring Abzena's strong innovation focus is translated into real-world solutions for partners. Abzena has carved out a distinct position in the CDMO and CRO landscape with a focus on complex modalities like antibody-drug conjugates and bioconjugates, underpinned by proprietary platforms such as ThyBridge and AbSelect Pro. In this teardown, we'll explore what makes Abzena unique in today's market, how they're addressing some of the toughest bottlenecks in biologics development, and where Campbell sees the biggest opportunities and risks on the horizon. Thank you for joining me, Campbell. Let's start with you then. Tell me a bit about your background, your career progression. Next week is your 10-year anniversary at Abzena. Congratulations for that. So tell me a bit about that and a bit before that. How did you get here?
Yeah, so how I got here, I'm a card-carrying immunologist, so scientifically I chose immunology because it was one of those, fantastically diverse subject areas that offered a lot of opportunity to get involved in potentially how to treat disease, because immunology is involved, the immune system is involved in so much. So that was the sort of scientific interest, but I joined biotech back in '97 in Cambridge in the UK, a group called Pharmaceuticals, and spent the next, best part of 19 years there, really truly understanding the kind of ability to take ideas from brain to bench to bedside, as I say, so translating great ideas into kind of great medicines to treat diseases. So spent a lot of time there. Loved the kind of energy, the ups and downs, the risks involved in trying to secure capital to take these ideas forward, the ups and downs of clinical successes, clinical failures. So it was a really dynamic sort of learning experience. But I brought that to Abzena, where Abzena was relatively early stages of building out an integrated service offering through early discovery, design of complex biologics, bioconjugates, and then building on from that towards the ability to manufacture those complex drugs. And so when I joined, incredibly exciting time. CEO at the time had a vision to pull together the capabilities that offered integration from concept to GMP manufacture. So we acquired a couple of sites out in the US where we focus very much on the manufacturing capabilities. One in San Diego for biologics manufacture, so process development, scale-up manufacture of biologics, and one in Bristol near Philadelphia, focus very much on the linker payload expertise, scale up GMP manufacture of that important component of ADCs, as well as the manufacture of the bioconjugate itself. And a lot of that, is underpinned by, developing and building out analytical bioassay formulation development capabilities. Because if I've learned anything in biotech, one of the fundamental critical things to make sure you do is generate data around the drugs you're developing, data to help select the best lead candidate to take forward, to inform how you might modify the design to remove any liabilities that are risks downstream for stability, scalability, safety, etc. But also, we built the business towards that kind of de-risking at the front end, with the science and technical expertise, and then moving that forward into the capabilities to scale that into manufacturing. And that, of course, is the second sort of aspect. It's great developing something that looks really attractive, and you've got the data then to attract investment, to convince regulators to take it to the clinic, and also, ultimately help our customers go through those value inflection points and help them to, say, raise capital. And we've got a bunch of examples of customers that have done that really successfully through Series A, Series B, they've IPO'd, they've out-licensed their drug, and or they've been acquired by pharma.
You've clearly staked out a position in complex modalities like ADCs and bioconjugates. What was the strategic logic behind concentrating there instead of chasing like monoclonal antibodies, for example?
Yeah, I mean, vanilla, there's loads of folks who can do vanilla antibodies extremely well. The usual suspects in the industry and others. But where we came from was that, as I say, that scientific, technical expertise, a lot of great brain juice, as I call it, around protein engineering, conjugation technologies, ADC formats. And so with that kind of level of expertise, it made sense for us to focus more. And we can do vanilla antibodies really well as well, but it made sense for us to focus more on these next generation biologics and bioconjugates. So we're seeing this increase and this growth in bispecifics, multispecifics, funky fusion proteins, as well as, moving on from the classical ADCs, which obviously that's still an area of significant growth, into, more novel bioconjugates such as antibody-oligoconjugates. Of course, we've seen the news recently, the weekend, with Avidity looking to be acquired by Novartis for $12 billion. And, we know Avidity. In fact, we've got a lot of experience with a lot of companies working in that space. And, I don't like saying we've ever got a USP or we're unique in any particular way, but I would say that when it comes to ADCs and AOCs in particular, we're at the forefront of the science and technology required to design and develop those and also manufacture those.
You said you don't like to say you've got a USP. How does that work in such a crowded environment? How do you approach that?
I mean, I think what sets us apart, whether you want to call that USP or not, what sets us apart, I would say, is a number of things. It's not just one thing. So there's the technology side of things. As I say, there's that deep scientific expertise that we bring to bear on helping identify and address and deliver on the challenges we face with the development of these complex new drugs. But also how we work with our customers. We're highly collaborative. It's not, we don't expect our customers to fit into how we work. We, as I mentioned earlier on, we understand their pain points. We understand what they need to do to go through those value inflection points, and we tailor our services and our expertise to fit into that. And we work with all sorts of different types of customer, those that are virtual, those that are, they've got their own sort of wet labs or their own, facilities somewhere, and even big pharma. So to expect customers to fit into, a cookie cutter type solution, especially, as I say, with this growth in the next generation biologics bioconjugates, that's not really going to work. So super technical, super collaborative, super agile, and we essentially dovetail into what our customers need. So if that's a USP, maybe, but it's certainly, I think, what makes us stand out from our competitors.
You're investing heavily in proprietary platforms like ThyBridge and Abzena Pro. How do you think about platforms, not just as science, but as business tools to shorten timelines, reduce risk, and build, long-time client retention?
Yeah, I mean, I think when you're developing drugs, there's a balance between, experience, expertise, what you've seen before, what works, what doesn't work, and then considering what kind of solutions you might want to offer. Now, oftentimes the solution is just being very good, very knowledgeable, high experts at the kind of area you're working in and you're offering services around. But also you can take that knowledge, expertise and apply it to developing technologies that actually provide solutions that address what we're seeing in terms of not necessarily the failures, but some of the weaknesses in the designs of these drugs as they go through the clinic and we see the data. So with ADCs, of course, there's a big issue of stability in vivo, toxicity, safety. So we've looked, using the brain juice within the business, the team have looked at how they can overcome that, and they've developed something called ThyBridge. And we've got a lot of preclinical data showing that this does what we expect it to do. So we generate a highly stable, highly uniform drug to antibody ratio form of the ADC. It's scalable, and it is something that we've seen performs better in vivo. Now, we've got one of our clients has taken this into the clinic, and we are eagerly anticipating data coming from that. But we have a strong feeling that it's going to do what we say it's going to do, and I think that we're going to see a better safety profile in many of the conjugate technologies that are out there just now. So being able to offer that as a solution, not this solution, we're agnostic, but if it fits what the customer is looking to do in terms of target product profile, then it's there to use. The other side of things is, the rapidity, being able to go from, idea to IND as quickly as possible, and de-risking where we can, because obviously if we work early on to identify risks and de-risk those, that's great. So we accelerate and we don't get caught in a kind of downward spiral of trying to figure out what's going on at the more expensive stage of development and manufacturing, or even once we see some safety issues, it's better to do that early. But producing the molecule is really important. So we've worked and invested in developing, best-in-class cell line development capabilities for producing the biologic. So we've used our own host cell line, which works extremely well with proteomics vector system, 2G unit vector system. And we found that fits into the ideal solution for our customers, because it's agnostic to the type of molecule that we put in there. So you see a lot of data around these platforms from all the folks that offer this, looking at antibody productivity. We've seen great productivity not just for antibodies, but with real funky fusions, real complex molecules. We're seeing the same levels of productivity there. It's highly stable, so it's as good as, and we, in terms of in our hands, it's better than the transposase data we've seen. It's another sort of sexy technology that's used. We don't use that. We use proteomic, and in our hands, we've seen that to outperform transposase in terms of, it's as quick as transposase in terms of timeline to get to a clone, but it outperforms it in terms of stability and productivity. So we're really pleased with what we've got, and that gives our customers an option to use that to make their material and go to manufacture, and confident that they're going to get the productivity they need and the stability in the line.
So I know you said, for example, your ThyroBridge platform, it might not be suitable for all of your clients' needs. How does it work there? A client comes to you, how do you ensure you've got the processes and technology set up to be able to serve them? Or, would there be a case where you might turn them away?
So that comes with the expertise in the business. In some ways, the business was founded around a conjugation expertise, as we call polypherics. So that is fundamental to what we offer as a company. That significant scientific expertise, technical experience. So the team have seen and done pretty much everything over 20 plus years that we've been in this business, in terms of linker architecture, payloads, bioconjugation technologies. So they're highly experienced at thinking about what's going to help with the optimal design, whether it's through lysines, cysteines, engineered cysteines, glycans, and marry that with what we expect from the target product profile, at least the mode of action expected of the cargo, the payload, and help our customers navigate through to get to that optimal design. So we don't push ThyroBridge onto them, but it might be a solution for them. But linking through glycans might be a better solution depending on the target product profile and the payload that they're using.
Can you share an example or case study where your approach has made a difference in kind of speed, quality, or outcome for the client?
Yeah, what's a little frustrating for us as a business, I guess, is that we have to maintain a lot of confidentiality around who we're working with, unless they're happy for us to prepare a press release and and make it publicly known that we're working with customers. But we have been seeing consistently over the last couple of years, outside of this sort of dry spell in investment in biotech, we've been seeing a lot of the customers we've been working with in the news in terms of successful points for them, whether it's IND, but in particular whether they've been acquired or whether they've out-licensed their drug for hundreds of millions or billions of dollars. That is really exciting. But one perhaps example, and unfortunately I can't say who it was, but one example that I think underpins who we are as a business was a group out in the US, an incubator, famous incubator in the US, group of 3 or 4 folks working on early proof of concept for next generation bioconjugate. We essentially took them out of that incubator and worked towards finalising the design of their novel bioconjugate, and we did everything around that. So we made the cell line for the fab piece. We helped optimise the manufacture process for their linker payload and bioconjugation as well, and scale up manufacture. All the cell line development, all the analytics, formulation development, and got them to IND in super quick time. Now in that journey, which was about from the early POC to clinic, it was around 2 years, which for a new generation molecule is exceptional. We had a great partner in that though. They were really aggressive and they wanted to do a lot of things in parallel, which helps. And they got the investment to do that, sort of blue chip VCs supporting them do that. They IPO'd before IND for a few $100 million, and they're worth multiple billions just now, and they're getting some really, really compelling and exciting data in the clinic, and they're looking to launch in the next couple of years. So that's what we can do for customers.
Yeah, that's really great to hear. I know your cell line development offer comes with very transparent pricing to IND and no royalties, which I think is unusual in this space. What's the strategy behind that model? Where do you kind of expect to earn that back?
I think first and foremost, having been at the other side of this in biotech for years, with cell line development, it seems to me to be a little bit smoke and mirrors. A lot of folks claiming, these megagrams per litre, and whether that's actually a benefit or not is questionable. But, we've got all these sort of data points that I think folks that are not experienced in the field, early biotech, see and respond to. But then, behind that there's what it costs, and then behind that there's what it costs to actually make the production cell line, and then behind that there's any license fees and milestones and royalties. So I think it's very difficult for customers to really navigate through that kind of very vague data points. So for us, it's important to keep it as clean as possible, as transparent as possible, so that customers know what they're paying, what they're going to get from the service side. Any license fees are minimal, highly competitive, and one-off. So we keep that so that there's no complexity around it and it's one and done. And then the customer can get on and deal with the bigger challenges of taking that molecule to the clinic.
Moving on, what specific bottlenecks in biologics and bioconjugates development do you think Abzena is really uniquely positioned to address? What about your portfolio really most excites you?
I mean, I guess it's part luck, part of the design, as we've kind of discussed. I mean, 20 or so years ago, who knew that ADCs and next generation bioconjugates were going to be so exciting, so successful? But that's what we built the business around, and right time, right place for us. And I think what we're finding now is, we see that at CPHI this week. We've got a lot of customers coming to us who are trying to get into this sector, into this field. They've maybe got an antibody portfolio. How can we help them get into ADCs? There's groups looking to get into AOCs as well. They've got really interesting targets beyond transferrin receptor, and they think that that's going to be super valuable to link siRNA or EOSs to that. So for us, as I say, right place, right time. Perhaps a challenge for us is to keep up with the demand, but it's a challenge that we're more than capable of delivering on. The other thing as well, it's maybe a little bit of a bottleneck, is from a geopolitical perspective. There's a lot of activity around kind of onshoring manufacturing in the US for obvious reasons. And again, we are right place, right time. Our manufacturing capabilities are out in the US, and the demand there is super exciting, and we need to respond to that because there's not that many manufacturers of ADCs in the US.
Absolutely. And how are you responding to that? Obviously, you have your site in Bristol in the US. Are you changing manufacturing kind of approaches there, or expanding or increasing portfolio there?
So there's two sort of aspects to this. One is the pressure our current customers are putting us under to go to phase 3 commercial. So on both our biologics and bioconjugate sites. So having that kind of validation and support, gives us impetus to grow. So we have in place now plans to get there very, very quickly in both sites. In fact, we've had a recent audit from one of our customers in San Diego, which went extremely favourable in terms of moving into that stage of development. We've also got space across the group that we can leverage in the US and think about optionality around moving conjugation around, particularly conjugation of the non-toxic payloads, AOCs, into some of our facilities where we've got that extra space and capacity to do so. So we've got a lot of flexibility within our business just now, but obviously we need to continue to look towards the future and what we need to do to keep up with the growth.
What do you measure to tell whether your strategy is working? What does kind of success look like for Abzena?
Yeah, I think it depends who you speak to. I mean, for me, I'm a scientist come drug developer, and I think that's one thing that stimulates our sort of technical team. They get to see, first on planet drugs that they actually help input in the success of, the design and manufacture of. So success is seeing these sometimes incredibly challenging, crazy ideas come to fruition when they get to patients. So that is a point of success. Obviously, we want to be able to maintain our kind of premier position in sense of being the premier group to come to for ADCs and AOCs and other novel bioconjugates. So we need to keep on top of technology, we need to keep on top of the capacity, as we just discussed. But, getting folks to IND, seeing them succeed in terms of, IPOing, raising capital to continue the growth of their businesses, which of course feeds back to the growth of our business and ultimately promotes improved health across, diseases where we never thought we'd be able to treat.
From your scientific perspective then, looking across the market, where do you see the strongest kind of demand and growth right now, whether it's in modalities, therapeutic areas, customer segments? If you had $100 million, where would you put it?
Bioconjugates, I think, is the future for the moment. This is the decade of ADCs, and with AOCs really cracking the nut in some of these undertreated diseases, I think we're going to see that grow into the next decade as well. I think what adds to that, we hear a lot about this around AI, and people saying, well, how are we applying AI to drug development? Obviously there's systems and processes that AI can apply to help improve the way we work in drug development. But in terms of the science and technology, we work with a bunch of companies who have AI platforms that are going to help identify new targets. So, while we're working on a lot of the old usual suspect targets and modifying the antibodies to improve outcome, whether that's making them into ADCs, whether that's using them in combination with, other treatments, with AI we're going to have a plethora of new targets that we can go after, which might be better, probably will be better than those usual suspects we're dealing with. So, between AI and the capability to design, these next generation drug types, I think there's There's a lot of legs in drug development as we go forward.
As a final note, obviously you have a big background in biotech. What one piece of advice would you give your younger self just starting out in that career on sort of manufacturing strategy and CDMOs, CROs?
I think it would be stay flexible, stay responsive, look to get that balance right between a platform that can be modified slightly to suit some of the different challenges that come along. We discussed a little bit of that with our cell line development platform, being able to accommodate, non-normal antibodies, non-normal recombinant proteins, and stay open-eyed and optimistic about the future, because I think, especially when you look through 22, 23, and the drop in investment in drug development and biotech, that can put a lot of people off. But if you've been around long enough, you know that it's going to come back. So being open-minded, flexible and agile and responsive, you're going to be in the right place at the right time.
That's great. Well, thanks a lot for your time, Campbell. It was great talking to you.